Greetings people, welcome back to Intoxication biology. The topic of today's long video will be proteins. As it was mentioned in the previous video, proteins are indeed the most common macromolecules found in living organisms. In fact, they are so common that even vegans and vegetarians cannot escape them from their diet. This is how a regular moderate sized protein looks like, and it is indeed very complex. Structural formulae of proteins always remind me of some subway maps of very complex subway systems, so like those found in really big cities, and to a certain extent it is really hard to memorize those structures, but still don't feel relieved. Because if your poor soul ends up in some biochemistry class, you will definitely have to memorize amino acids and those really complex names of proteins. Obviously, proteins have a lot of different functions. In the metaphor of organism as a society with those different people and classes, proteins would be definitely found in almost every class. Like for instance, white-collar digestive enzymes packed into the cubicles of duodenum or any other part of the gastrointestinal system. And then there are defensive enzymes such as leukocytes, white blood cells, and macrophages who protect the body from pathogens. Proteins have their proper representation in the body's Illuminatis, such as the proteins which control endocrine system and genes, like for example insulin, which controls sugar, and the transcription factors which would control the process of gene transcription. Receptors that spread information throughout the body to necessary parts, they are also proteins, by the way. And finally, the working class of proteins, the structural proteins such as collagen, elastin, keratin, which all provide structure in different places. For example, keratin provides structure for hair and skin, elastin is responsible for elasticity of tissues, while collagen is a major component of the connective tissue. Proteins are polymers, which means that they are composed of really similar chemical structures. As it was discussed in the previous video, the amino acid, which is the component of protein, is composed of amino group, a carboxyl group, the mysterious R group, hydrogen group, and all of that is attached to the carbon, which is in this case named alpha carbon. Like alpha male, alpha carbon gets all of the attention and all of the 4 bonds in the amino acid. When you add up all those amino acids in a long chain, you get this polypeptide chain, which has respectively the amino terminal or the N terminal and the C terminal, which is also the carboxylic terminal. Since polypeptide has amino group and acidic group, it is both base and acid at the same time. Another interesting feature of amino acid is that it can have 2 isomers, 2 optical isomers. One of them is oriented at the right and the other one is oriented at the left. In real world, however, only left-oriented amino acids exist, or also known as L-amino acid. In such a way, this world is all governed by really left, almost communist amino acids. Polypeptide bond becomes polypeptide camaraderie. So perhaps the easiest way to memorize those amino acids is by first of all grouping them into 5 major groups. In general, there are positively charged amino acid, negatively charged amino acid, polar amino acids, and nonpolar amino acids. And finally, there are just special cases of amino acids. There are 3 things that you may need to memorize for amino acids. It may be the name of amino acid with 3 letters, the name of amino acid with 1 letter, or the structural formula of amino acid, which is probably the hardest thing to memorize. The best way to memorize perhaps would be to connect all those obscure names and structural formulas to certain associations and certain things which are not really related to biology. So when you think about the positively charged amino acids, try to think from the perspective of China in case of Hong Kong when it was returned to China from Great Britain back in 1997. So it's definitely a very positive moment. So that's where you have the association with positively charged amino acids. So HKR, Hong Kong return, stands for the first one-letter abbreviations of each amino acid in the positively charged amino acid group. Also, you can memorize the three-letter abbreviations for those amino acids by thinking of, first of all, history, because this event involves British and Chinese history. Also, lysis. Lysis is breakup, so there was a breakup between Hong Kong and China and breakup between Hong Kong and Britain, whichever way you prefer to think of it. And also there was a certain argument between two sides, and finally this argument was resolved. Now moving on to the structural formula of histidine, which is really complex. So if you memorize this, it's a really big thing. So first of all, history is complex and it repeats in a loop. So it means that there is a loop inside this structural formula. Also, when you think of the alpha carbon, you have to think of the cordon sanitaire, an additional carbon which will separate the alpha carbon from the loop itself. So because alpha carbon doesn't want to have this loop close to it, so that's why it requires at least one carbon between them. So an easy way to memorize the loop is as following. So think of the last 5 presidents of the United States and think of the respective parties which were represented. So in the case when you had a Republican president, you insert carbon. In case when you have a Democratic president, you insert nitrogen. So we have Reagan and we have Bush older, both carbons. We have Clinton nitrogen, we have Bush younger carbon, and we have Obama nitrogen. Pretty simple. And also, once again, remember the order of the specific elements in the loop. Also, important point about the loop is that you have to memorize the double bonds. You have to memorize that there's a double bond between Bush and Reagan, and there's a double bond between Bush and Clinton. So basically, each Bush has its own double bond with previous president. So in case of lysine, the structural formula is pretty simple. So since lys sounds like lice, in this case you can think of the 4 extra carbons which are required to separate the alpha carbon from the lousy amino acid. And also 4 is considered to be unlucky number in Hong Kong or China or whatever. So in this case, it's really easy to memorize that you require specifically 4 carbons. And finally, there is arginine, which is associated with argument. So when you think of arginine, think of an argument between alpha carbon and another carbon. In this case, 2 carbons are so angry at each other, so they decided to separate each other with 4 monomers. And 3 of them are just plain carbon groups, and one of them, the closest to another C, is just another amino. And also, since C is kind of fearing the attack from the alpha carbon, it decides to create this surrounding of 3 amino acids, and one of those amino acids actually forms a double bond with carbon. So then we have negatively charged amino acids, and when you think of negatively charged amino acids, think of Deutschland. Even though it may be a good country, think of Germany really negatively in this context. So try to think of Holocaust, Nazis, whatever else you want to insert, since you have to memorize this structure of D and E for aspartic and glutamic acid. Also, for the 3-letter abbreviations, you have to think of asp, which is a venomous snake, and glue. In this case, something negative about glue would be thinking about glue as a substance to abuse or to eat or to do some other things which can result in very bad consequences. Memorization of negatively charged amino acids is really simple. Aspartic acid structure is very similar to glutamic acid structure. So for aspartic acid, just think of carboxyl group separated by one CH2. In this case, just think of carboxyl as something negative, and in this case, alpha carbon just wants to separate from it with 1 extra carbon. So in glutamic acid, the separation increases to 2 carbons, but everything else stays the same. You have the same carboxyl group. Next we have polar amino acids, and when you think about polar amino acids, think about something cold, because usually the north and south pole are pretty cold. In this case, for the memorization of 1-letter abbreviations, you just think of the mnemonic Y question, where you just omit all of the vowels and you get the Y question. Also, you will have to memorize the 3-letter abbreviations. In this case, think of some things which are related to coldness and to something located at the north. So in this case, we have winter tires, we have threshold, which is somehow related to temperature going beyond the threshold. We have also a snowflake, and Glenn and serial killer are not really related to coldness and to north. However, you can think of Glenn as a specifically natural thing which occurs only in the north, and for serial killer, well, in this case, think of the cold-blooded mindset of serial killer, which is quite kind of related to the coldness. So let's examine the structure of tyrosine, for example. In this case, once again, we have carbon, alpha carbon, which doesn't want to connect itself to the major structural compounds, such as benzene ring in this case, which represents the winter tire, and also we have a hydroxyl group at the end of benzene ring, which can represent the antifreeze, which is also used often in the winter time. So once again, alpha carbon separates itself from all of those with an extra carbon, and hydroxyl is located at the most extreme point far away from the alpha carbon, since antifreeze is really dangerous for alpha carbon. Next we have glutamine, which is associated with Glen, and Glen is associated with nature. So every time we have something natural and related to nature, we have to realize that there is an amide involved. So here it is very simple. We have 2 carbon separation and a simple amide quite far from alpha carbon. So serine is associated with serial killer, and in this case serial killer is methanol, because methanol poisoning is quite common in cases when people try to substitute for ethanol. And here methanol is just simply attached to alpha carbon, nothing special. So here we have threonine. In this case we have to remember of threshold, and here we have a very interesting configuration of ethanol. So it's not a usual configuration of ethanol, but in this case we have hydroxyl very close to alpha carbon, and it shows that alpha carbon is actually very interested in alcohol group. So once again, in order to relate threshold to threonine, just think of the importance of knowing the ethanol tolerance threshold. Next we have asparagine, which is related to a snowflake, and in this case it is also related to nature, so we have an amide. But here amide is separated from alpha carbon only by 1 extra carbon group. Next we move to our largest group, which is the nonpolar amino acids. In order to memorize the 1-letter abbreviations of nonpolar amino acids, think of Volkswagen and family. So family Volkswagen in this case. And when we move to our 3-letter designations of amino acids, we have to think of something which is opposite to polarity, and thus it is opposite to coldness, and we think of something which is hot. So first of all we have phenomenal for phenylalanine. We have also alanine for Los Alamos. So here Los Alamos was located in New Mexico, and it was also site of nuclear testing, so it all involves very hot things. Meteorite is also quite hot. Ilium is also a very hot place, since digestion needs some temperature. Leukemia is kind of related to Los Alamos, even though it's not related to hotness. Still think of it in the category of nonpolar amino acids. Valparaiso is a city in Chile, so this is quite evident that you will have some degree of hotness. And finally, there is therapy for tryptophan, and it's related to trip to some equatorial country and some vacation in warm places. Okay, so alanine is really simple. If you need to memorize alanine and you have some problems with that, well, I'm really sorry for you in this case, because it's only a simple carbon attached to alpha carbon. So for phenylalanine, it is slightly complicated. So phenylalanine is just an extension of alanine. In this case of alanine, let's think of atomic bomb testing, and in case of phenylalanine, let's think of something more powerful like hydrogen bomb testing. So in this case we have benzene ring attached to that carbon, and that makes testing of Los Alamos even more phenomenal. Next we have methionine, which is associated with meteorite, and in this case sulfur plays the role of meteorite since it falls right into the carbon skeleton, but as usually it falls very far from alpha carbon, but still it disconnects the carbon skeleton. So the best location for that is between the second and the third carbons in the carbon chain. Next we have isoleucine, which is composed of a very simple 4-carbon chain. In this case, this 4-carbon chain looks like a human intestine, where you have appendix or cecum, whatever pleases your guts, and then we have 3 carbons which represent the 3 parts of small intestine, respectively duodenum, jejunum, and ileum. And they diverge at the point where the 3rd carbon meets alpha carbon. So then we have leucine, which is associated with leukemia, which is associated with radiation. And in this case, we have a simple isobutane that looks exactly like a radioactive hazard sign, which is also pretty much like a logo of Mercedes. So in order to memorize valine, you don't really have to think of Valparaiso because that one was just for memorizing that it has to do with nonpolar acids. In this case, just think of the initial letter V, and you have just an inverted V sign or Asian peace sign if you're into that. So here we have tryptophan, and tryptophan is a very special amino acid since it has a very special function. Its function is to torture the minds of those people who have to memorize it. So here we have an extra carbon that separates 2 loops from alpha carbon. Then we have the larger loop which is far away. It's a benzene. Just memorize the structure of benzene. It is very simple. For the smaller loop, you have to be really specific. So first of all, there are double bonds that are never together. Also, there is 1 nitrogen that never has double bond, and 1 double bond that is part of the benzene. So if you memorize those simple rules specifically for tryptophan, you will actually memorize the greater structure of it. And finally, we have special amino acids where we have a very interesting mnemonic to memorize: the German Communist Party. I'm not even sure that such party exists in Germany, and that's why it makes it special. So here you have the connection with special amino acids. For the 3-letter abbreviations of special amino acids, think of the following words: glycerin, cyst, and pro. All of those things are quite special. So glycerin is pretty special kind of soap. Cyst is a pretty special thing to form on human body, and pro is also quite special in a good way, of course. So glycine is the most famous amino acid since it's the favorite amino acid of all slackers. Usually when they ask you to memorize only 1 amino acid for upcoming test, everyone will memorize this, which is quite pathetic. For cysteine it's also quite simple. Just look at C and S, the 2 consonants in the 3-letter abbreviation of cysteine. So it stands for carbon and sulfur. Just plug it in and you got the structural formula of cysteine. And the last amino acid is a very special amino acid, since the R group of this amino acid just completely brutalizes and destroys the amino group by intervening right into it. So this is a very special amino acid called proline or proline. But proline is better to memorize since it's actually not a real line but rather curve. So this is a very deceptive name. And once again, there is a modified amino acid group, and also make sure that you memorize that there are 3 carbons in the skeleton, which is part of the propane. If proteins had a religion, most likely their religion would be Hinduism, since proteins also have this similar concept of 4 stages of life. Life of protein is very simple: primary, secondary, tertiary, and quaternary structures. That's it. The primary structure of protein is simply like the early childhood, when protein accepts and grows in length by forming this long polypeptide chain, which is linear, simple, and basic. But however, in the secondary stage, which signifies the identity crisis of protein, there are 2 choices that protein can take. Either it becomes an alpha helix or beta pleated sheet. However, similar to this cat that has an identity crisis of confusing itself with a lizard, proteins can actually embrace two identities at the same time. They just have different segments of alpha helix and beta plated sheets in their structure. With additional folding at the tertiary stage, the mature and real life of protein begins. Folding and bending occurs due to hydrogen bonds and disulfide bridges, which are like long-lasting friendships. Hydrophobic amino acids aggregate together into large groups where they share their fear of water and polar substances. Some intense, passionate ionic bonds form between electrically charged amino acids. And suddenly there comes the quaternary structure, which is nothing bad because protein still exists and it just reunites together with other proteins into a larger and greater and more powerful protein. Also, it's important to note that all those interesting bonds formed in tertiary and secondary structure and in quaternary structure as well can be actually destroyed or altered or modified by the environmental effects on proteins, such as the presence of polar substances, the presence of non-polar substances, increased or decreased pH, and of course temperature. And everyone knows this pickup line about denaturation, that hotness denatures proteins. Anyway, denaturation can be reversible and irreversible. Reversible degeneration was shown in a Finsen experiment where the possibility of reconstruction of proteins was proven through increase and decrease of pH. Of course, denaturation is not something very good for organisms because in this case when proteins cease to function, the organism also ceases to function. So for this case, there are special things called chaperone proteins. Chaperone proteins protect other proteins from denaturation, and probably you get the idea from the picture. So chaperone proteins are very similar to the real chaperones at the different parties and dances, which protect the chastity and the virginity of protein from the detrimental and vicious effects of environment. Chaperone proteins were discovered first in 1962 by Italian geneticist Ritosa, who conducted a very strange experiment by putting flies into a heated chamber, which could be a microwave actually. Anyway, such case of horrible insect abuse resulted in discovery of heat shock proteins, which are responsible for preservation of different important proteins from the temperature shock. So that's it for today. Hopefully you survived those 20 minutes of protein torture, and the next video will be on carbohydrates and lipids.